The overall goal of this procedure is to collect phloem exudate from Arabidopsis or other plants to analyze its contents or confirm the presence of compounds within the phloem stream. This is accomplished by first cutting and incubating the petiole in potassium EDTA to prevent sealing of the sieve element. The second step is to thoroughly wash the petiole to remove any remaining EDTA that can interfere with the subsequent analysis and also damage cells during long-term exposure. Next, the phloem exudates are collected into water. The final step is to prepare samples for subsequent analysis by LCMS, GCMS, gel electrophoresis, and thin layer chromatography. Ultimately, the EDTA-Facilitated phloem exudation can be used to analyze and confirm phloem contents like metabolites and lipids, proteins, or RNAs.
Plants cannot escape adverse conditions. As a result, they require very efficient and fast systems to transmit environmental signals throughout the plants and respond by changing their development. One of these signaling pathways is the plant phloem, which is quite complex. To understand its composition, we use the EDTA-Facilitated phloem exudation for the collection and analysis of phloem contents. It was originally developed in Perilla by King and Seward, but can easily be modified for other species. Demonstrating the procedure will be Olena and Urs, both undergrad students from my lab.
Prior to starting this procedure, grow Arabidopsis plants for 4 to 6 weeks.